since we were talking a lot about therapeutics. So to put this in perspective, how does genomics impact on therapeutics? We're very different from one another, as you heard already, and our diseases are often very different. So pharmacogenomics refers to matching drugs to your genomic content, your DNA sequence. And basically, many diseases have very specific markers. So I think one of the best things we've learned from the genomics projects is that diseases that were thought to be the same are actually quite heterogeneous. So leukemia, I mentioned earlier, there are many forms of leukemia which you can separate out from genomic markers and RNA expression that you would never see just by looking at the cells. And the same is true for many other diseases as well. You can start separating that out with genomic markers. The other thing to point out with regards to background is that most people don't respond to drugs either because the drugs are not targeting what they should be targeting, or maybe because the drugs are not being effective in the patients because they're not at the right concentrations and such. And so really genetic factors, just to jump to the bottom of the slide, they can affect the drug concentration in the body, Stuart mentioned this, and how active a drug is against its target. So we have enzymes in our body that control drug metabolism, and many of these are actually sex-specific, meaning that they vary between men and men women don't respond to drugs the same way quite frequently because we have different, we're not built this way for the drugs, we're built this way because we make hormones differently, but nonetheless it has consequences on drugs. And so Stuart mentioned this, so I won't spend much time, but actually this is a classic example. This is an anticoagulant used after heart attacks and other cases, and basically there are two genes whose, they're both functioning, but they function at different levels, and depending which combination you have determines whether you're sensitive, whether you will metabolize this drug quickly, in which case you're fairly resistant to it, or if you don't metabolize this quickly, you'll be fairly sensitive. So Stuart, you must be fairly sensitive. So these drug metabolizing enzymes have been correlated with this, and if you can envision that one would like to match all of these different drug metabolizing enzymes essentially with all drugs. That hasn't been done, but it's actually in some respects a fairly straightforward project. And then one could actually get reasonably specific dosing of drugs. And again, you would match it whether you're a man or a woman. Okay. The other type is which is matching drugs to disease. And the classic case of this is a particular marker that's on breast cancer. So people are now finding specific markers on specific cancer. It gets at the question you were raising. And the classic example is HER2, which is a receptor that's expressed on a subset of breast cancers. It's anywhere from 25% to 50%, depending on the study you look at, are HER2 positive. And people who are HER2 positive, which you test by a very simple antibody stain, will be responsive to therapies that use antibodies against this receptor. So basically these patients get treated. If you're HER2 positive, you will get a HER2 antibody, and you'll be responsive to that, meaning your cancer will regress to varying degrees. It's not a perfect cure, that's for sure, but it does have significant effects on survival rates. Perhaps the most classic drug that's out there is a drug called Iressa, and that's unfortunately what got lost here. But this is a drug used to treat non-small cell lung carcinoma, and it was discovered somewhere near about 5 years ago that about 10% of patients who have non-small cell lung carcinoma will respond to this, amazingly so. That is, they virtually get cured of this, although resistance will form usually 5 or more years later for a subset of patients. But nonetheless, if you're a responder to this, I should point out when this is diagnosed, it's almost always diagnosed late. So if you find this, it's usually at the 11th hour. And as I say, one of the treatments is to treat patients with Iressa. But it turns out it only works if the patients have very specific mutation in a receptor on the surface. So that is to say, the individuals who have this mutation will actually get cured, whereas the other 90% is completely ineffective. It turns out the drug, if you see it binding to the protein there, if it actually binds and stimulates the cells to kill themselves. It's a really cool mechanism which they figured out. I mean, I have a personal story with this. I had a cousin fairly young who was diagnosed with this. And again, it was very, very late, and he had he was eligible for just a limited number of therapies, one of which was Iressa. He was a non-responder, and actually he passed away. And that's the value of cancer. You usually, if you diagnose these things really late, you only have a very small window in many cases in which to try a treatment, and you can't afford too many misses. And so that's why getting your therapies right away can be a matter of life or death. And so knowing how these work. So there's not that many examples of these, quite frankly. There's a limited number of antibody drugs that can be matched against this sort of thing. But there's enormous promise. As we learn more about the cancers and the specific subtypes that are out there, the expectation is we can design therapies. My own bias is that RNAi is actually fairly difficult because of the accessibility problem that was raised earlier. People tend to go after cell surface markers for this sort of thing because that is accessible. And antibody therapies tend to be fairly popular for cancer these days because they can bring in the, as David pointed out, they can actually help bring in the immune system, actually cure the cancer. So this is a very popular route. You're going to see zillions, antibodies, by the way. People thought when monoclonal antibodies, these are specific antibodies, came out a few years ago, everybody thought this is the cat's meow, this is going to cure lots of diseases. They went out of favor because people are using mouse antibodies and things. And they've only come back recently in the last few years because they're now humanized and our body recognizes them and can deal with them properly. And they're now coming back with a roar. You're seeing lots and lots of antibody therapies out there. Yeah.
Question about either this or rapid genome sequencing giving you blood tests for cancer early onset cancers. What's your prediction for that?
Yeah, my own view is that cancers are very heterogeneous, and what we're finding is that they seem to be activating pathways. I think David alluded to this, rather than necessarily specific targets. So if we can see the pathway signature that's activated, then you can try and design a therapy to this.
Not therapy, I'm talking about early detection.
Oh, for early
when
detection.
A cancer cell breaks out into the blood flow and you're able to pick it up and actually either sequence it or get an antibody pickup, so you can get a small cell carcinoma, lung cell blood test that you pick up early on. So
right.
You get scanned yearly basis for a panel of 500
Well, this gets
cancers.
us back to what I was saying earlier about I think we need incredibly sensitive diagnostics against lots of markers, because I don't think any one marker is. So I think this is, in my mind, this is where proteomics and metabolomics and such should step in. And I think that's what we need as part of routine blood testing, to be profiling for thousands of things so we can try and find these. My own view, we have another thing we've been doing in our lab. Your immune system is a very good sensor of things that are going wrong. So we've actually been looking for what are called autoantibodies against various disease states, ovarian cancer, asthma, things like that. And there are antibodies out there. So we're hoping that that might be an early diagnostic signature, but it remains to be seen if it's early enough. I mean, nobody really, it's a great question you're asking, and I don't think we know the perfect answer to that.
One technology you might want to look at, just real quickly, one technology you might want to look at is one of the groups that was funded by Stand Up to Cancer is a Harvard-MIT collaboration that's looking at a new methodology for picking out circulating tumor cells. And they think they can pull out one tumor cell out of a billion. They have a little device that has nanoposts on it, and it's got the appropriate antibodies on it to then recognize the cell surface proteins from a tumor cell as distinct from normal blood cells. And one of the problems they have is getting a few cells out of a reasonable blood draw and then actually washing them off without any contaminants and figuring out what's going on with those cells. The downstream analysis is becoming an issue.
Mike, yes. You mentioned earlier that you were working on this mass spec technology to do basically a proteomic analysis on blood. How much time will it be before we'll have, let's say, a test of a few $100s that can look at hundreds or thousands of analytes floating around in there?
At some level, we can do that now. I mean, the technology is coming online. But it's a matter, the problem is, I think a lot of what we want to look at is the really rare stuff in the blood. And those tests are not reliable enough just yet. So we want to find, the complication with blood is which is different from many other things, is that the dynamic range is extraordinary of the components in it. It's 10 to the 12th. People say for very important components that are needed, like a lot of the cytokines will be 10 to the minus 12 relative to the most abundant things in the blood. And so most people think that a lot of the markers we'd be interested would be in that very rare class. So we need to be able to reliably detect those things, and I think that's the key. So we're not reliable. The technology's there, we're not reliable next. I think within a few years we will be there, and I'm hopeful actually that antibody measures will be a good way to reliably find these sorts of things, but that remains to be seen. So I don't think we're far, and then I hope that that will be a powerful diagnostic tool for these things, and then you can sort cells. Once you know what to look for, you can sort them to find them and do the genomic analyses you'd like. Okay. Should I just say, I'll just say one word on microbes, or you want to blow over it?
So
all right. The one word is that microbes are everywhere. They're good ones and they're bad ones. You actually have 5 pounds of microbes in your gut. And that's good stuff, generally. It's helping you digest your food. Yeah, right. You want to taking antibiotics. It's true, you will lose weight. And microbes are, I mean, a lot of them are bacteria. There are other sorts of things too. So the word I want to give you is that a lot of people are trying to look at the microbes in the body and your gut and your skin as diagnostic tools as well. So I just wanted to throw that out there. And then just for thinking about the future. And then bad ones, I mean, we need measures for following bad ones, and this is really a huge area to look at. And I don't need to say much. You all have heard of H1N1 and have may or may not have done something about it, but that's a clear case of there's always emerging diseases, and we need lots of tools for getting at that. And that's all I'll say then. Is that all right, Stuart?
Mm-hmm.
Oh.
Yeah.
One.
What's the economic model for pharmacogenomics as the potential pool for these drug companies gets smaller and smaller down to single individuals? Why should they bother developing?
Yeah, there's two problems with this model. One is that no pharma company wants to make a diagnostic to go along with it, so that's a pain in the butt. And the other is no one wants to stratify their market, right, as you were just implying. The problem is that they may have to, because as these diseases are recognized as heterogeneous, as the marketplace gets crowded against some of the same areas like breast cancer and stuff, if you're not first in the market, you better be better. And one way to be better is to target a very specific population. So that may be the economic reason. And there's still a lot enough. It's tricky because you want to always hit $1 billion a year drug. Nobody would make a product for less than that. But some of these markets are sufficiently large that you might be able to have 2 or 3 players in that market. I mean, the statins are a good case, right, with Lipitor and things of that sort. So there are multiple players. But in general, you're right. I mean, there is disincentive. It's mostly an incentive if you're number 2 into the market.
So that's it with scary health stuff. We're going to now go into personalized traits. When I think about my human genetics, I think more about traits, a little bit less about medicine and health. It just so happened that I read this news article. There's a company called Ancestry.com. It's a company that tells you about your history. So you go onto the website, you load up your name, and they might tell you who your great-great-great-grandparents were, where you came from, and when you landed in the US, and all sorts of long-lost relatives. The newspaper article was about them going public. So they went and raised $100 million the day they went public. So I started thinking about this, and then what I want to tell you now is I think that there's a way better way to do Ancestry.com with your DNA. And so the idea is that, so Ancestry.com follows you by your birth record and by the census, and it kind of figures out by your name and the names of your parents and the names of their parents that, you know, like the Kims coming from a certain region, well, some of them might have been my relative. But a much better way to do that is with your DNA, because the idea is that each of you is barcoded with your DNA.
You realize Kim is hopeless.
Kim is hopeless. 40% of all Koreans are named Kim. So, and Seoul, Korea has, what, 10 million people? So Mike and I were at this meeting. It was a riot. There was a talk by this person from Korea. And she gave a really nice talk, I thought, about sequencing a person. And at the acknowledgement slide, it was like 30 people, but they were all named Kim. And so it's going to, I'd like to thank Kim, 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 and 3 more Kims over here and 4 more Kims over there. And she said, I thought it was great. But the idea is that each of you has 3 billion pieces of DNA, actually 6 billion pieces of DNA because you have 2 copies. And when you were made, there's something like 200 errors were put into you for the first time in the history of the world. So these are called spontaneous mutations. They occurred in you. They don't happen in your mother or your father. So think of it as you sequence your mother, you sequence your father, you look at you, and almost all of you come from your mother or your father except for 200 spots in your genome where it's a brand new change. Okay? So I want to call that a barcode. Now those changes are so infrequent, there is nobody else in the world that has those same changes as you. There's probably nobody else in the world that has even one of those changes the same as you. They're so rare, all right? So the idea is that your children are the only children in the world, or in the history of the world, who has those mutations, because you are the first ones to have those mutations, all right? Now you can turn it backwards. So here's my little experiment. Here's a person that was born in 1000 AD, some random person. And that person, for the first time, had these 200 little tick marks, which are point mutations in that person's DNA. So there was Queen of Scotland or something like that. And they had 2 kids, brother and a sister. And the brother had these barcode was inherited because now once the barcode is made in that person, it gets transmitted to both both son and daughter. But now the son has his own set of point mutations put into him. Daughter has her own set of point mutations put into her. So this one's barcoded and this one's barcoded. Well, that goes on for 50 generations. And every one of those people is barcoded with a new set of DNA mutations. And now you get to us today. So here, Mike sequences all of us. And there's something like 100 million sequences running around. Most of us in this room will be sequenced in the pretty near future. So I'm going to look at all my sequences, and I'm going to see the reference sequence. And I'm going to see these point mutations that nobody else has. So all of you guys upload your sequence, and I upload my sequence, and I have something that's never been seen before in anybody else. So I think that was in my lineage, and that either I had it or my father had it or something like that. But it was pretty private, okay? So is that clear? Now, if I see that I have this red mutation, if I had this blue mutation, and David Haussler has this blue mutation, it would be pretty weird for us to share. But we could know that we had pieces of DNA that came from this person in 1000 AD, because this person in 1000 AD is the only person that had that type of mutation. The fancy name for that is identity by descent. Okay? So David and I would be identical by descent in this region of our DNA. So actually, it's a whole clump of DNA. So if we had this whole clump of DNA and this whole clump of DNA, and David and I had all of these little private mutations, then David and I would probably know that we were derived from this person and this son of that person. All right? So I think this works. And that in the future, where there's a large number of people sequenced, I'm just going to look and see who has the same very, very rare alleles, and that will identify somebody that I was related to. Okay, that's complicated. It can't be. You guys are nodding, but it's complicated. All right, so, all right, here's Ancestry.com. Yeah.
So those 200 mutations, where does that information come from?
That's the DNA sequence. The question was, those 200 mutations, where do they come from? So when I was born, well, when my father was making sperm, my mother was making her egg, that process was a little bit error prone. And there's something like 200 mistakes made when my father was copying his DNA to make the sperm. And those 200 errors got put into me, okay? And that's just the mathematics of it, that the replication of the DNA is pretty good, but it's not perfect, okay? So the empirical numbers, there's 200 changes in you and in me.
How many of the original 200 mutations from, say, 1,000 years ago do you expect to survive anywhere in the population today?
Good question.
So how long are you going to have that signal around for?
So that is the exact same question. The question is whether those mutations survive.
They won't. I mean, if you just pick somebody from 1000 AD and look at those mutations, most of them won't. But if you run the process backwards, you're going to have these, and they must have come from somewhere. And so, by definition, all of these rare things that are segregating came from somebody.
So,
right.
So you have to think about this process going backwards.
No, I don't. Okay, hang on a minute. So I don't know if I understand that. So I think that in 1000 AD there was something like 100 million people in the world. I'll make that up. And now there's 6 billion. So on the average, every piece of DNA that was there in 1000 AD has to be replicated and present someplace on this world now some number of times because the population has grown.
No, the overwhelming tendency, because you only pass on half the copies to any given child, there's an overwhelming tendency for things to disappear after many generations.
I see. So the answer to your question is basically the pieces of that person's DNA that were passed on to daughter, son, or granddaughter, grandson, all of those pieces of DNA that exist presently will be found. Okay? And what David is saying is that there's a chance that some of the DNA is lost at every generation, and if it's lost ever from everybody, then it's gone. So if you only have one kid, half your DNA is gone. Okay? Yeah.
Like last names, yes.
Okay. So that's a very good point. So the math of this is complicated. It's way complicated. But the concept of this, I think, is valid. So this is the idea behind, yeah. Yell. I'll repeat your question.
So there, just to try to figure out if there's 6 billion people and 6 billion base pairs, then if there's 200 mutations, would it really only be one allele that would make the difference, or would you need all 200 to look at the combination? You know what I'm saying?
Yeah, I know what you're saying.
Yeah.
So you have 6 billion base pairs, and each of them can change to 3 types of things. So, and there's only 6 billion people in the world. So that's the math. The answer is that there aren't very many people that are going to have the exact same point mutation, even one. And then to have all 200 that you have, I think there'll be nobody. Okay? Now, so this is my last slide on this. So this is Ancestry.com. And here I am. That's not me. That's a woman. But if I was a woman, here I would be. And these are my brothers. I would know that. I'd know my mom and dad. I'd know my uncles and aunts. So these are my aunts. These are my uncles. I'd probably know my grandparents. And I might know my great-grandparents. But then, who knows if the birth records start to peter out or something bad happened. And Ancestry.com is trying to do it this way by following names and birth records and being really good about that sort of thing. But it's encoded in your DNA. And so I think that the day that Mike gets his 100 million genomes sequenced, you don't have to do this anymore. It'll be barcoded. And that Ancestry.com will be swallowed by 23andMe because 23andMe will be able to just look this up. Yeah. And when they say Ancestry, they'll mean Ancestry. So I'll have, this is today. I think in the near future, I think I'll have thousands of generations. I'll be able to know, oh, I didn't tell you. This works theoretically for a lot longer than what I told you. I just made up 1000 AD. This works to old times. It works to ancient times. It works to prehistory. So somebody, I think, 10,000 years ago that was born, that happened to be the person that I inherited DNA from, I still have that point mutation from that person, and I could know it. So if David and I were related by the same caveman 10,000 years ago, we could possibly know that. So that's going to, I think, just blow 20.
But only in that part of the genome.
Only that part of the genome.
You have to be clear on this.
And you still have to ask, you know, how the hell did that happen? But thousands of generations, this will work. Not 1000 AD is like 50 generations ago. This will work for a very, very long time. And if you look for thousands of generations, you can get millions of relatives. So I'll get, you know, I happen to know most of my first cousins, but now I'll know, you know, my cousin's 100th removed if I wanted to. So here's a case where, you know, I think there'll be science. I don't actually know what you do with this. I asked my son, you know, I told my son, I tried to bounce this idea off you. I said, I think this would work. And he'd say, well, who cares? Okay? And then I said, well, who cares? But then I read the newspaper article that said that silly company made $100 million. Somebody cares about finding more relatives. So maybe there's a... so I am going to talk about aging. So I study aging a lot in my lab. And here's what I want to tell you about. There's this company called Surtris. Surtris is, one of the co-founders is Christoph Westphal. And here's his quote: in 5 or 6 or 7 years, there will be drugs that prolong longevity. So I think that's really amazing. And I actually happen to agree with him. I don't know if it's 5 or 6 or 7 years, but I think for us, we will have the chance to seriously think about whether or not we want to live longer, which is one of those ethical questions. But scientifically, I think it's possible. So here's Surtris. Surtris was made based on this, this discovery by David Sinclair. This scientist and this businessman got together. They were bought out by GlaxoSmithKline for $700 million after about 5 years. Here's the discovery, the kind of discovery that was made. So in 1995, scientists had found a couple of genes that can make things live longer. So here's an example of a thing that makes a worm live longer. This is a normal worm that dies with this kind of kinetics. So here's the worm living, and then it's dying as it gets up to about day 25. And then you get a mutation in the worm, and it can, you know, one kind of mutation lets it live this longer, and another kind of treatment lets it live this longer, and can live a lot longer. Turns out the same gene that makes the worm live longer, if you do this, it makes a fly live longer, and it makes a mouse live longer. And now there's evidence that this same gene makes people live longer. So that's what David Sinclair worked on, another kind of gene, which is shown here. And in 1995, there were basically 2 genes. One was called the insulin receptor. This one was called the SIR2 gene. And this chemical here, resverotrol, activates the SIR2 protein. And when you give it to mice, this is a normal mouse. And if you give it to mice, the mice live longer with resveratrol. So that was the idea, that in the late 90s, they had figured out a gene that makes things live longer called SIR2. Makes worms live longer, just a minute, flies live longer, mice live longer. And they found a chemical that activates the gene, which could make things live longer. And the chemical caused worms to live longer, flies to live longer, mice to live longer. Started Surtris, and then a few years after that, they've got something that's 100 times more powerful chemical that they're putting into phase 1 and phase 2 trials for people and to see if that. My whole point is that in 2009, there's hundreds of genes that make animals live longer, model organisms live longer. So in 1995, there were 2 hits. Now in 2009, there's 100 hits. And maybe you don't have to be a super dynamo like Christoph and a super smart guy like David Sinclair. You could be a regular guy, and you could probably recapitulate the progress of Surtris and designing targeted pharmaceuticals that can activate, inactivate proteins that make you live longer, at least in all the model organisms and probably in humans. The issue is then when you make a human live longer, how do you get that FDA approved? So Surtris is not making longevity drugs. They're making anti-diabetes drugs because when you live longer, then your insulin sensitivity gets way better. So the whole point I'm trying to say is that scientifically, it seems like the pathway to become the next Sertris is wide open. There should be many, many companies that could recapitulate Sertris based on the next gene or the next gene or the next gene that seems to make animals live longer. Did I answer your question?
I figured the chart out, sorry.
Okay.
Quick question. When you compare flies, worms, mice, do you find that the longer lived the animal is, the less of an increase there is in the lifespan with these things? Or is it?
No. So the question was whether the drug or the gene works better on the short-lived animals than the long-lived animals, and it's the same percent. It's like 30%, 50% increased lifespan. And although there are some genes that cause 100% increase in lifespan in worms, and there's combinations of genes that can get over 100% lifespan.
Do you think humans, given that there's a similar percentage,
right?
Probably we would expect a human to
doubling,
similar 30%,
doubling.
Well, doubling in that other case, but in the case of resveratrol, maybe 30%.
Yeah. So the question was, how good will this work in humans? So, you know, if I could buy 10 more year lifespan, that would be a big deal. But for a person that works in the aging field, 10% increase in lifespan is nothing. You know, we're shooting for, right now my lab cutoff is 50% lifespan. Otherwise, we don't want to study it. But it's not that hard for us to get 50% increase in lifespan. So, you know, my lab has 3 different things that we're tracking that do 50%. And that's just the beginning.
Stuart, the question is at what point is this with resveratrol being given at start of life and throughout at midpoint?
No, this is adult. And so the question is, do you have to have it when you're young, or is it, can we still do it, you know? And so those are the key scientific questions. What the hell's the answer for resveratrol? I think the answer is that, I know with resveratrol it was adult mice that had it. I don't know if they did just young mice or old mice, you know, could you, and when was the best time in your life to take it? But that's the key question. Okay. I really want to just do this athleticism thing in 2 minutes just because it's funnier than heck. So I got together with a colleague who used to play in the NFL and 23andMe who gave us 100 gene chips, and we called up 100 NFL players and genotyped them and compared them to the rest of us to do this, and then ESPN wrote an article on all of this stuff and a little TV show. But the idea was to compare elite athletes versus normal guys to see what was so good about being big and strong and tough and rough against us normal guys. So that was good. So we get to genotype all of these people. And then the answer is that there was not one difference between the NFL players and the normal people. Either that, or we don't have, with just 100 people, we didn't get to see enough of a statistical difference and it wasn't a big enough experiment. But if we did 30,000 NFL players, maybe there would be. But it wasn't screaming. Only 1 in 300,000 people play in the NFL, although every single male in the US wants to play in the NFL, only 1 in the 300,000 does play in the NFL. So it's a tale of a population that was genotyped of the roughest, biggest, tallest guys. I thought worked pretty well. Better than that is that I became sort of like the genetic counselor for 8 of the NFL players. So I looked at they emailed me their genotype, and I looked at all of their genes. And I had looked at about 50 different genes that had some sort of evidence from prior studies from the literature of being different between athletes, elite athletes and normal athletes. And that was kind of interesting because there you don't need a complete 16-fold effect for Alzheimer's or breast cancers or things like that. A tiny little edge. These are guys that make $4 million a year if they start. And if they're second string or if they get cut, they get very little. So they really want to start. And the difference between starter and non-starter can be very small. So any tiny edge in how good they are or not getting hurt was big. So that worked pretty well because, you know, I talked to tight end for the New Orleans Saint. And the tight end had this he was homozygous for a rare allele of collagen 3A. And that rare allele is associated with less ACL injuries. So he's walking around playing this incredibly dangerous sport for his life, for his occupation, but he has knowledge that maybe he's not, he has a little bit of an edge in not getting hurt, this very serious knee injury. So that could have been useful for him in training. It could have been useful for him in negotiating his contract or with his coach. If his agent could have known that, that could have helped convince the team that he was the one that wasn't going to get hurt after 1 year. And it could have helped him know whether he should have played to 34, 35 when he's not that healthy anymore, but he's making a lot more money than he's ever had just because he's established his name. But he was wondering about whether he would be walking at age 50. So this same allele that protects you for ACL injury also protects you from osteoarthritis. So when you get older, you'd like to know if you're being protected. So I think that, outside of the box of just health and that, the general population, what do they want to know about? I think they might want to know about something like their athleticism, if they could either track their athleticism or track how similar they are to different athletes that might get genotyped. That might be fun to do. David.
Okay, so real fast, everybody's obsessed with sequencing people, but having this wonderful technology allows us to look at how actually all of the different life forms on this planet evolved from common ancestors. And most people are most interested in what are sometimes called charismatic megafauna, the animals that we know and love and see in the zoo. Mammals is one big group of those. So imagine with genome sequencing technology, you could sequence something from each of the mammals. I'm showing just a few out of the 5,000 different placental mammal species here. You could start with the humans. We have that sequence. Svante Pavo's group is going to give us a sequence of the Neanderthal pretty soon. And when we compare that and compare it to outgroups like chimpanzee and bonobo, we'll be able to see kind of what our genome of our ancestor looked like about 700,000 years ago. As we go back, we see the 98.3% similar genome of the chimpanzee. We're going to get the bonobo as well. And by comparing those, we can understand what the common genome looked like 5 million years ago and what's changed. So we can have a list of genetic changes that happened on the human lineage and happened on the chimpanzee lineage, and that's enough to take you from the common ancestor to these species today. And this just keeps going back. We can actually compare genomes and deduce what common segments look like and what changes happened. All the way back through the placentals, and I'm just taking you on a quick tour. There are 14 steps where you could reconstruct an ancestral genome that spawned some lineage. Here we are back to the Boreoeutheria, and then after 100 million years, you reach back to somebody that was shrew-like themselves. So here's an animal that looks more or less more like the ancestor than many others. And you would be able to then recapitulate these genomic changes going forward in time through these 14 steps to get a human, or if you took a different path, you would end up with something possibly very different, like an elephant or a porpoise or a bat. So what are the genetic changes that underlie the spectacular evolutionary innovations that occurred with these large animals as the vertebrates conquered land, air, and sea? One of the projects that I'm leading, hoping to push on that, is we made a proposal just a few days ago for planning for a project to sequence the genomes of 10,000 different vertebrate species. There's about 60,000, but we could understand better their divergences. We could understand the vulnerabilities of rare and endangered species, and they do have genetic vulnerabilities, and we could basically launch on one of the most exciting scientific missions I can imagine, really figuring out how all of these different species actually did get here in terms of their genetic changes. So that's it. Social and ethical issues were things that we didn't have time to go into in detail, but there are, of course, a number of social and ethical issues involved in all of this. And this is the last slide.
Great. Are there any questions? Comments?
that allow it to differentiate.
You want it, that's epigenetics. When you sequence stem cells, you don't want to just sequence the DNA. They'll have the same DNA as the other cells until you get to cancer and immune.
Your question was, can you figure out what makes a stem cell versus a differentiated cell?
Yeah.
And so people, I think that's not in the DNA sequence. That's in particular regulators that make the stem cell give it its stemness. You know, and a really, really exciting breakthrough was just a couple of years ago where they found a set of 4 transcription factors that seem to cause cells to become stem cells. And you can take skin cells, put in these 4 transcription factors, and they become stem cells. That's called an induced stem cell. So now I think a reasonable thing for everybody to do is take some of their skin cells, put in these 4 transcription factors, and then save it away as your own stem cells. So that if you get a disease, I get a disease, I can put in my own stem cells, hopefully to cure me of those diseases. Peter?
So if you guys had to put your finger on, I'd love answers from each of you, the top 3 breakthroughs in the next 5 years we should be watching out for, that in your area is sort of like the Holy Grail. What would they be? And when do you guess they're going to happen?
I'll start. Single cell, a genome from a single cell will be
a complete genome from a single cell.
A complete genome from a single cell will be an
that's
enormous breakthrough. 10 years.
10 years. What, another one?
We can.
Okay,
I can share the wealth there.
I'm going to go with aging. So I think I want to be able to eat something, take a drug, or do something, or I'm going to double my lifespan.
And time frame, Stuart?
My lifetime. 10 years.
10 years. Very cool.
I think a gradual process is what we're seeing already, the tailored design therapies for a wide range of diseases, and that'll be ongoing for the next 15 years. If there were one landmark thing, I do think this issue about correcting your IPS, getting IPS cells, correcting them and fixing yourself, either through organ replacement or other various degenerative diseases in particular, that'll be pretty soon.
30 years from now, what's going to knock a person out? I mean, are we basically going to have knocked down most cancers, most diseases, and most ailments?
Probably car accidents and infectious diseases. I would worry about infectious diseases. You know, think what would have happened if SARS were, or sorry, H1N1 or SARS. First of all, it's amazing SARS got controlled if you think about it. But H1N1, imagine that's mortality rate goes up tenfold. That will certainly have a strong effect. It already is having a significant effect, even at its, by biological standards, a rather modest mortality rate.
And let me ask another question to you guys then. When do you expect we're going to start seeing the first designer babies and or sort of human redesign happening?
Well, the germline stuff will probably be a ways off, I think, because for lots of reasons, ethical amongst them and safety. Somatic changes, I think you're starting to see some already. So ADA is one area that's been corrected, although not so strongly, but then there's been some recent ones on blindness. I don't know if it's sleepers or lepers, eye degeneration. So that will come along at some level. And I think, you know, right now gene therapy is a therapy of last resort. And I think it'll stay that way for a while. But at some point it will kick in.
Peter, I think you ask a really profound question. So I think we attained self-knowledge in 2001 when we sequenced our DNA. And so we're the only animal that might not have to evolve. We could design, right? So right now it's not ethical. But someday, well, because most of the things we have won't work and we don't know how to design ourselves. But someday, you're going to actually know how to improve people. Somebody's going to say, well, this, not only theoretically, but I'm pretty damn sure this is going to be an improvement. And then the question is, why or who? I mean, it might not be you or me because I'm not going to do it. But some future world, it's just going to be so obvious that you can improve somebody. Why not improve somebody that way instead of some other way? So that's our future. That's not the near-term future. That's space travel and time travel and things like that in the future. We'll be able to design people.
But there's an interesting point at which it goes from is it ethically right to is it morally an obligation if you have the ability to do something. I might posit the point that you give the best you can to your children in education, in clothes, in everything you can, and if you have the option,
right?
Would you find that an obligation? We're going from a species of evolution by natural selection to evolution by intelligent direction,
right?
So to speak.
I buy that.
Intelligent design.
I didn't say that.
Don't use the
I specifically said intelligent direction.
I think we're going to go through a long period of increasingly being able to select which alleles we pass on, which of our own alleles we pass on to our children. And that's going to create a quite tumultuous situation within the society itself. But that's very different from inventing new alleles out of pure cloth and more or less designing the baby from scratch.
One of the things that we do here is we talk about the intersection of all these technologies. So at the same time, we're talking about robotics and human-machine interface and space travel and such. And all of these things are coming online in the next 20, 30 years, brain-machine interface and such. It may well be that we need to, meeting in the middle is genetically engineering the interfaces that will connect to the appropriate non-biological interfaces, whether it's increased resistance to radiation if you're traveling to colonize Mars, or whether it's better interfaces to your nervous system if you're trying to interface Google up in the cortex.
Right.
So.